Welcome to Introductory Animal Physiology, Week 5, Lecture 4, that is Introduction and Microbial Fermentation under the module Ruminant Digestion. I am Dr. Tulasi Raman, Assistant Professor in Veterinary Physiology and Biochemistry at the Faculty of Veterinary Animal Sciences, Institute of Agricultural Sciences, Banaras Hindu University. In this session, we will explore the complex yet fascinating process of microbial fermentation in ruminants. This lecture will set the stage for a deeper understanding of how ruminants efficiently utilize complex plant materials through a highly specialized digestive system. Let's embark on this journey through the ruminant digestive landscape, examining each process in detail. So ruminants, as I say ruminants, why they have given the name as a ruminant? There are certain animals which are called as ruminants just because they possess rumen in them and then they does rumination. So because of this reason, some animals are called as ruminants. The ruminants stomach, if you take, there are two compartments. One compartment is a non-secretory part and the another compartment is secretory compartment. So the non-secretory compartment is called as forestomach, whereas the secretory compartment of ruminants is abomasum, that is true stomach in the ruminants. The forestomach, if you take in the ruminants, it is comprised of rumen, reticulum and omasum. So if you see the overview of the ruminant digestion is that the ruminant will intake in terms of feed or food. As it takes the feed or food, that will goes to the rumen where it will acted upon by the microbes where the fermentation will takes place. You know that the prerequisite for fermentation to takes place is an anaerobic environment. So under the anaerobic environment, the microbial fermentation will takes place by simply hydrolysis process in the presence of carbon dioxide and it generates the volatile fatty acids. There are three important volatile fatty acids produced in the rumen is that propionic acid, acetic acid and then butyric acid. So these volatile fatty acids is used to generate ATP in ruminants, but this is not the case in the non-ruminants or a simple stomach animal. Now the animals are classified based on the diet as well as the digestive physiology. First, if we see with the diet point of view, the animals are classified into herbivores, then carnivores and then omnivores. This you know very well. Herbivores means these are the animals which ingest the plant materials. So these herbivores are broadly classified as ruminants, and then non-ruminants are, you can say, monogastric. Monogastric means single stomach animal. Also are a simple stomach animals, you can say. Let us come to the classification of animals based on the digestive physiology. It is classified into ruminants and then non-ruminants based on the physiology, digestive physiology. So the example for ruminant is that cattle, that is cow, buffalo, sheep and goat, deer, elk, antelopes, kangaroo and then giraffe. So these are the animals which are example for the ruminant animals. Whereas coming to the non-ruminants example, you can say pig, poultry, dog and cat, horse and mule, rabbit, zebra, rhinoceros, elephant and rodents. In addition to that, other animals also are there, many animals. In this, the horse, mule, rabbit, zebra, rhinoceros, elephant, rodent and then camel. Some places they used to mention it as pseudo-ruminant. Why they call it as pseudo-ruminant? Because these are the species where they are not having all the compartments which should be possessed by the ruminant animal, such as rumen, reticulum, omasum and abomasum. Instead, these animals will have only three compartments or three parts in it. One is forestomach, and then midgut, and then hindgut. So these are the three parts we can say. That is why these are the animals which sometimes called as a pseudo-ruminants. Because, and another reason is that why it is a pseudo-ruminant or it is classified under non-ruminant is that because they won't ruminate. So due to these reasons, this will be called as a non-ruminant or a pseudo-ruminants. Now, in this lecture, in digestive system of ruminants, we'll see first is the dietary substrates, then the fermentation environment requirements, what are the advantages of a ruminant being a ruminant, and what are the disadvantages of being a ruminant in the digestion. Then overview of ruminal microbes, rumen digesta, then fermentation of dietary carbohydrates, then dietary protein, and then dietary lipids we will see in detail. First is that dietary substrates. So the substrates is broadly classified in animals, the feed or food you can say. One is roughage and the next one is concentrates. So the example for roughage is that fiber materials which is rich in fiber, that is example you can say grasses, different grasses. So these roughages are nothing but it is a form of long fibers of cellulose embedded in the matrix of hemicellulose. See here, cellulose and hemicellulose. These are the keywords here. Now, these cellulose, these are nothing but a beta 1,4 glucose linked polysaccharide. Right? Whereas, this is you can say in other words, it is a hexose, example of hexose. Then whereas hemicellulose, these are beta 1,4 linked xylose linked polysaccharide. You can say this is a pentose. Okay, so one I have given the hexose, another one is a pentose. Now, to digest these hexoses and pentoses, that is beta 1,4 linked, either it is glucose linked or it is a xylose linked, it requires a special enzymes such as cellulase, which is required to digest the cellulose. Then hemicellulase, lase means it is when you mention an enzyme, it will end with ase, A-S-E. So hemicellulase is an enzyme is required to digest the hemicellulose. Then pectin lyase. This is an enzyme is required to beta 1,4 linked pectin linked polysaccharides. And then fructanases. So this is an enzyme which is required to hydrolyze the fructase, right? So these are the enzymes which are necessary to hydrolyze the beta 1,4 linked polysaccharides. These enzymes are present in microbes, or it is synthesized by microbes, and it is also present in the plants. So due to this reason, a ruminant must have a symbiotic relationship with the microbes. So this is one of the important prerequisites to be a ruminant. So that is why the herbivores, if I say a ruminant, that means it should establish, the animal should establish a symbiotic microbial fermentative relationship with the microbes. Okay, this is very, very important, symbiotic fermentative relationship. Now, the next is that grain or concentrates. When we say the grain or concentrates, these are nothing but alpha 1,4 linked or alpha 1,6 linked. Okay. Now, to hydrolyze the alpha 1,4 linked or alpha 1,6 linked polysaccharides, it requires an enzyme amylases. Okay. Now, this amylases is present in the alimentary tract, or it is produced, it is by the animals as well as microbes and plants. So because of this reason, even the non-ruminants are having amylase. Okay. Whereas in ruminants, in addition to the animal, even the microbes as well as the plants contains the amylase enzyme that is required for hydrolyzing the beta 1,4 linked polysaccharide as well as the alpha 1,4 linked polysaccharide and alpha 1,6 linked polysaccharide. So this is about the dietary substrates. Now, where the fermentation, when I say fermentation, the fermentation happens at, based on the site of fermentations, the animals are classified as foregut fermenters and the hindgut fermenters. Okay, based on the site of fermentation, again the animals are classified as foregut fermenters and hindgut fermenters. Foregut fermenters means, fore means front. Okay, that means gut before the stomach, that is a foregut. Hindgut means, hind means back. So it is after the stomach, that is hindgut fermenters. That means the fermentation takes place after the gut, the stomach part. Whereas there are certain animals where the fermentation takes place before the stomach, that is a foregut fermenter animal. Example for foregut is that ruminants and then camelids, the camels, right? And then hindgut fermenters, horse, rabbit, rat, guinea pig and swine. So this is about the classification of animals and little bit introduction about the dietary substrates as well as the classification of animals based on the diet, digestive physiology and then site of fermentation. Now, the next aspect in the digestive physiology is that in the ruminant digestion is how being a ruminant, it is having some advantages as well as disadvantages. So the advantages of a ruminant, being a ruminant, is that first, it can able to utilize the diets that may be too fibrous, which cannot be done by the non-ruminants. So that is why the grass materials can be ingested and then digested and ultimately get absorbed also the nutrients. So because of that reason, the ruminants are the first important advantage is that one. So utilization of the diets which are too much fibrous in nature. The next is that ability to break down the cellulose and related compounds. As I told in the earlier slide, cellulase, hemicellulase, these are some of the structural carbohydrates. You can say from that you will get this type of compounds. So the polysaccharides, so cellulose, hemicellulose, pectins, right? So these are all the compounds which require to hydrolyze, it requires an enzyme, and that enzyme is produced by the microbes. So that is why the symbiotic relationship is required, that I already told. So these enzymes are required to break down the cellulose and its related compounds. And this is possible only in the ruminants, not in the non-ruminants. Then that means monogastric animal, I can say. Then the third important advantage is that being a ruminant is that synthesis of high biological value microbial protein. Many a times, whatever the animal ingests, in non-ruminants or monogastric animals, directly the dietary proteins will be taken from that, the proteins will be hydrolyzed and it will be absorbed. Whereas in the ruminants, this is not the case. It will use to take the plant materials. So in the proteins which is present in the diet is of low quality or poor quality. So hence these proteins will be taken by the microbes and it will get converted into a high biological value microbial protein by integrating it into the cell wall. So this synthesis of high biological value protein, that means the conversion of a low biological value protein into a high biological value microbial protein, that will be easily digested and get absorbed by the ruminants. That is beneficial for them. The next is that simply if you supply the cobalt is sufficient to synthesize the vitamin B12, that is cyanocobalamin, that is sufficient to synthesize the vitamin B complex by the microbes with the help of the microbes. So these are some of the advantages I discussed of being a ruminant animal. Then what are the disadvantages of being a ruminant? The first and foremost is that it is a voracious eater. So at regular interval, it requires food. That is one important disadvantage. The next important disadvantage is that it almost spends 8 hours per day in simply rumination or gut chewing. Rumination, you might have seen when a cow sits ideally without having anything, that time it will try to bring back the content from the stomach part. That means the rumen to the mouth, where it will just chew again, and then so that the larger particles get broken down into smaller, and then it will again ingested. The deglutition will take place. So the same way, almost 8 hours per day it try to spend for the just for rumination or the gut chewing process. That is what it's spending large parts of a day. Then in order the fermentation vat, the rumen is a fermentation vat. Vat means it's nothing but a vessel. So this rumen is acting as a fermentation vat. In order to work efficiently, this vat, there are certain requirements are there, such as regular addition of a large quantity of alkaline saliva, because the saliva of ruminant is rich in bicarbonate ion. So that will act as a buffer in order to neutralize the acids produced in the rumen, such as a result of the fermentation, such as volatile fatty acids. And then powerful mixing movement of the digesta is required, then only the proper digestion will be achieved. And then mechanism for elimination of the gases, eructation. As a result of this fermentation, there are huge amount of carbon dioxide as well as the methane and other gases used to produce in the rumen. These gases must be eliminated or burped at regular intervals. So this is another important disadvantage. The next is that the fibrous material will leave the ruminoreticulum area until it reaches the maximum size of 2 to 4 millimeter in length. Okay, till that time it will stay in the rumen itself or the reticulum itself. So it will keep on digesting, and it will try to break down into a smaller particle size, which is a maximum of 2 to 4 millimeter in length. Then, unlike non-ruminants or the monogastric species, the pathways of the intermediary metabolism is totally related with the end product of the volatile fatty acids, whereas this is not the case with the monogastric intermediary metabolism pathways. So the intermediary metabolism pathway of the ruminant is entirely different. Then the simpler monosaccharides as well as disaccharides which is present in the diet is mostly utilized by the rumen microbes. As you may think that these ruminants are not having a capability to produce glucose. No, it is having a capability to produce glucose like the non-ruminants or the monogastric species. But these glucose are taken by are ingested by the microbes for its survival. On other hand, these microbes helps in the release of the enzymes which is required to digest the fibrous material, as well as it able to produce a high biological value microbial protein. That is how the symbiotic relationship is existing, right? So that is how it is acting as an energy source. Then fermentation environment requirement. Now the fermentation to takes place, there are certain requirements are there. Already now you know what are the advantages and disadvantages. From that itself you can understand what are the requirements, right? But still we'll see here one by one. As I said, fermentation, it will takes place. The first important requisite is the anaerobic condition. The carbon dioxide, the redox potential is very, very, very important. Then the next is that regular addition of new and macerated substances, the chewed food, is required. Otherwise, what happens? It may not able to mix, or the enzymes may not act properly, or the microbial population may not be maintained. So due to that reason, the regular addition of the new and the macerated substrates is required. Then presence of a suitable number of microbes as well as the type of microbes. We'll see what are the different types of microbes. There are many types of microbes are present in the rumen, such as bacteria, fungi, and protozoa, in addition to the other microbes. So the suitable number of bacteria are the microbes, as well as which type of microbe is required for particular diet, so that the digestion can be achieved at its maximum limit. Then the steady removal of the end products of fermentation, because if it won't takes place, then what will happen? There is no space to accommodate the new products to be formed, the end products. So as a result, it will stay there itself. So due to that reason, the end product of the fermentation as well as the digested materials must be removed regularly from the rumen so that the new products can be formed, right? So that is why steady removal of end products of fermentation, because that will act as an inhibitor of fermentation because there is no space to accommodate more end products. Then the mixing and addition of new substrates and the buffers. So this I told in terms of volatile fatty acids as well as in terms of the substrates. So in both these cases, it has to be eliminated at regular intervals or it should be moved from rumen to the next compartment. Otherwise, the space is not sufficient and so that the new products or the new substrates may not be digested properly. The enzymes may not act. Then dispose of unfermented materials to abomasum and intestine. When there is some undigested material is left in the rumen, it has to be regularly removed. Otherwise, what happens? It will keep on get accumulated in the rumen, and as a result, the new substrates or the substrates which are coming after the chewing the gut, it may not be digested and absorbed maximum. Then the maintenance of the intraruminal conditions such as temperature, pH, osmotic pressure, and the redox potential. These are highly essential for the fermentation to take place at proper. Then let us come to the ruminal microbes. So these are some of the requirements for fermentation to take place. Next we'll see the ruminal microbes. The ruminal microbes is of three types broadly. First is that bacteria, then second is that yeast-like fungi, and the third is that protozoa. If you see the microbes, the bacteria are further classified into primary bacteria and then secondary bacteria. So primary bacteria, like example, you can say the amylolytic and cellulolytic bacteria. So amylolytic bacteria means what? Amylo, that is related with the amylase. Lytic means it is lysine, right? So these are the bacteria which is required to hydrolyze the amylose in the ingested feed. Okay, so that type of bacteria are amylolytic bacteria. Then cellulolytic bacteria, these are the bacteria which are essential to hydrolyze the cellulose compound which is present in the food or the feed. You can say the cellulolytic includes the hemicelluloids and the other compounds also. Then the rumen bacteria also classified based on the environmental existence as well as how it is utilizing the substrates and end products it produced. So depending on that also the bacteria can be classified. So first is that environmental existence. Based on that total 5 groups of bacteria. The bacteria are classified into 5 groups. First is that the bacteria which are freely living, that is associated with the rumen liquid phase. So there are different phases are there in the rumen. In the digestive, next slide we'll see. So in the liquid phase, these are the bacteria which are freely living, freely survival. Okay. Then next is that the bacteria which are loosely associated with the feed particles. Next is the bacteria which is associated loosely with the feed particles. Then next classification is that bacteria which is associated firmly with the feed particles. Then the fourth one is that bacteria associated with the epithelium of the rumen. Okay. And the last one is that the bacteria which is attached with the other microbes such as protozoa as well as fungal. So these are the other classification of the bacteria. Then the next is that based on the what substrate it is utilizing and which end product it forms. Based on that, you can say cellulolytic, which I already told you, it's a primary bacteria, hemicellulolytic bacteria, pectinolytic bacteria, amylolytic bacteria, ureolytic bacteria, methane producing, sugar utilizing, acid utilizing, proteolytic, ammonia producing, lipid utilizing species. So these many types of bacteria can be classified based on the which type of substrate it utilizes and which end product it produces. Based on that, it is classified. So example I would like to give for some bacteria, like for example cellulolytic, is that the first one is that ruminococcus. The ruminococcus species, you can say, is an example of a major cellulolytic bacteria. Like that, we can say there are many bacteria, many examples are there. Next, we'll go to the protozoa. So the protozoa is of different types. Among them is the most abundant one are entodynomorphs as well as holotrichs. So these are the most common protozoa present in the rumen, right? So in addition to these endodynomorphs and then the holotrichs, it also having epidymium, then isotrichia species, then dasytricha species, then diplodyne species. So there are different types of protozoas are there. Among them I told already, that is endodynomorphs, and then holotrichs is the most abundant ciliates are present in the protozoa. Then what is the importance of the protozoa? The protozoa is an indicator organism. What it indicates? It indicates the rumen healthiness. Okay, because there is a slight change in the microenvironment in the rumen that directly affects the protozoal count. So that is why we can say it is a very good indicator to say the healthiness of a rumen. And then another important aspect of the protozoa is that this is the one and only reservoir which is there to provide a PUFA, that is polyunsaturated fatty acids, to the ruminants. This is the one and only source, and that's how it is acting as a reservoir for PUFA as well as other substances such as starch. Then there is a way to classify these microbes based on substrates, is that the protozoa starch degraders, soluble sugar utilizers, lignocellulose hydrolyzers. So in this way also you can classify the protozoa. Now we'll see the major microbes in the rumen. The first one is that the protozoa, ciliate protozoa, which is present in huge numbers, followed by non-spore forming anaerobic bacteria, then followed by anaerobic fungi, and the last one is that facultative anaerobic bacteria. So this is the composition based on the numbers, right? Let us see the overview of the rumen digesta. The rumen digesta have 4 phases. The first one is the dense liquid phase, then second one is a solid phase, and the third one is a liquid phase, and the fourth one is a gaseous phase. So gaseous phase, mostly I told carbon dioxide, methane, so these are present, right? So it has to be removed at regular intervals through burping process. Then coming to the liquid phase, where the fibrous material, which are in nature large and light particles, used to float in the liquid phase. Whereas coming to the solid phase, in that the hay and grain materials of yesterday's in nature, right? And because these hays or grains already it stayed more than 24 hours and it broken down into smaller particles. That is why you can say in this solid phase you will find the smaller and dense particles, you can say. Fine. Then the last one is dense liquid, where all the debris used to settle at the bottom because of the heaviness, and it has to be eliminated, removed. So the next important aspect here I forgotten is that pH, right? Rumen always stays healthy when the pH is above 5.7. This is very, very important. So the protozoa, primary cellulolytic bacteria, and the most secondary bacteria used to survive well in the pH of 6.2 to 6.9. Whereas the primary amylolytic bacteria, which is active when the pH is approximately 5.8. Remember this. And when there is a lactacidosis condition, in that case the pH decreases, and the number of microbes, the quantity and quality, it varies depending on the pH of the rumen. So the protozoa usually will be plenty when there is a starch rich diet is provided to an animal. So with that, I am what I discussed is that we have seen what is a ruminant. First of all, we have classified the animals based on the diet, digestive physiology, and then the site of fermentation. Then we saw the dietary substrates, that is roughage and the concentrates. Then what are the fermentation environment requirement. Then we have seen the advantages and its disadvantages of being a ruminant. Then overview of the ruminal microbes. Then overview of ruminal digester. So with that, today's lecture is ending. Thank you.